Dear all, today I am going to tell you about non-invasive source of stem cells. Stem cells from urine. Variety of stem cells are available from urine, but two different and significant cells from urine sample are urine stem cells and renal cells. These are undifferentiated and donor-derived cells. Most important is urine stem cell or USC. This is the progenitor cell and it can be converted into multiple lineages. This is derived from kidney and this is confirmed due to the presence of high expression of kidney cortex markers. Compared to renal cells, these generate more donor-specific multipotent cells by cell culture. Renal cells, these are less potent than USC in gene expression and in vitro expandability. Culture medium for both type of cells. Here, first of all, for USC, 10% fetal bovine serum in Delbeco's modified eagle medium, nutrient mixture F-12, and keratinocyte serum free medium with supplements. This is the culture medium, and these cells have been isolated from patients of few diseased conditions. As for renal cells, these cells require renal epithelial cell growth factor, and that is used in the culture to grow these cells. In this case, uh, these are always produced or obtained from healthy individuals. There is no data available for these varieties of this variety of cells from diseased patients. After isolation and expansion, both cell types can be treated with dimethyl sulfoxide DMSF and stored in liquid nitrogen, and they hold a strong revival capacity for future application without considerable loss of viability. This means both of these varieties of cells can be expanded and then they can be stored once they are treated with dimethyl sulfoxide, and storage is done in liquid nitrogen, and from research it is clear that very less loss occurs in number of cells when they are revived to renew a culture. Differentiation capacity of urine derived cells and their potential application. This table shows details of different qualities. Here first type is urine stem cells, which is able to differentiate into endothelial, uroepithelial, smooth muscle, myogenic, beta cell-like structures, cells, osteogenic, neuronal, chondrogenic, and induced pluripotent stem cells. If it is endothelial, urine markers expressed before differentiation. This is these two markers are prominently found before differentiation, and after differentiation these are the markers which show this phenotype, and it has been tested in vivo also. Its potential application is renal construction, angiogenesis (SUI), and erectile dysfunction. Then again, this urine stem cell can be differentiated into uroepithelial cells and marker expressed before differentiation is uroplakin 1A, and after differentiation or trans differentiation, markers are uroplakin 3 AE1, AE3, and CK7. Because of the presence of these markers, these are known as uroepithelial cells, and these are also tested for in vivo testing. Urological reconstruction is the potential application when these are differentiated into uroepithelial cells. Then they can be differentiated into smooth muscles, and one marker expressed before differentiation is alpha SMA, and after this differentiation, desmin, myosin, smoothulin. These are the variety of markers which show the presence of smooth muscle cells, and it is also tested in vivo conditions uh, and utilized for bladder reconstruction and genitourinary repair. Myogenic, when it is differentiated to myogenic cells, then no marker was there previously, and afterwards MyoD, myogenin, myF5, myosin. These are the uh, markers. Uh, these markers were present on cells, and this was also used for in vivo testing for heart repair and SUI. Beta-like cells, again, no marker was previously present, but after differentiation, PTXF1 is the marker which is characterized uh, for beta-like cells, and it was also reported that in diabetic treatment, this type of differentiated cells have potential. Osteogenic differentiation marker was nil, and after differentiation, osteocalcin, RNX2, and ALP. These markers were present, and it was also used for in vivo testing, and here bone tissue engineering was done utilizing this type of urine stem cells. Neuronal cell lines are also created, and for that previously previously shown marker was SOX2, and afterwards it was there were many markers showing the presence of neuronal cell. These are GFAP, nestin, NF200, S100, but it is not uh, tested in vivo, and potential है कि it may be used for neural tissue engineering. Then chondrogenic marker was not, any such marker was not present since the beginning, but afterwards SOX9, collagen 2, agrican, these are the markers which showed presence of chondrogenic nature of these cells, and it is tested in vivo also for cartilage replacement. Now these cells can be converted into induced pluripotent stem cells, and previously present markers were KLF4, SOX2, OX3 and 4, CMYC, and afterwards they showed NANOG marker. And in vivo testing में it, 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 it was tested in vivo also, and it developed teratoma. When these cells are converted into or differentiated into iPS, teratoma formation was found. And it has potential application in disease modeling and drug screening. Now, renal cells, these can be differentiated into neural cell, beta cell, iPS, induced pluripotent stem cells. Original markers present in the first case were SOX2, but afterwards nestin and PAX6 was present, showing the presence of neural stem cells, and it has been tested for in vivo also, and it has potential in neurodegenerative disorders. Beta cells, they have some markers present since very beginning. Before differentiation, SOX17, PTX1, while after differentiation, NKX6.1, insulin, and C-peptide. These three are predominant markers for beta cells, and it has been tested for uh, diabetic therapy, and in vivo testing has been reported. Now it it can also be converted into induced pluripotent cells, stem cells, and markers. Any differentiation marker is not reported, so we can say unknown. While afterwards, after differentiation, SOX2, OC3, 4, KLF4, TRA1, CISA. So these are the markers which show pluripotency, and in vivo testing has been done with the development of teratoma, and it can be used for disease modeling and drug screening. Teratoma, because of the formation of teratoma, and now these cells are in this form, these cells are less useful. Now this table has been concluded. Sorry. Urine stem cells has been shown to generate differentiated cells for kidney, genitourinary uh, part, cartilage, bone, and cardiac repair. Renal cells have been utilized to generate differentiated cells such as beta cells and liver cells. Pluripotent stem cells have been generated from both renal cells and urine stem cells. This is the summary of this table, and now this is again a flow chart of utilization of urine cells, stem cells. Here any donor can be present to donate his or her cell, or it is patient for autologous treatment. Then urine is collected, it is isolated and expanded. Then expansion may be done uh, by automated techniques or by cell culture techniques later on. This can be used directly from this stage, and it can be differentiated by uh, use of different growth factors, or so that a particular type of cell is made, and then uh, this is given to patient, or through indirect differentiation, means here these are converted first into induced pluripotent cells, then these are differentiated and used for drug screening or tissue engineering. टिश्यू इंजीनियरिंग मीन्स कि stem cells are utilized for making some tissue, and that tissue is used for replacement therapy, or that is utilized for making any organ. Conclusion of this, that statistical analysis suggests that using the daily average volume of urine from an individual, 10 times the number of donor specific urine cells required for an autologous tissue repair can be generated. Means sufficient cells can be generated. Then automation of urine cell cultures will make it easy to achieve numbers for human transplantation. Here, थिंग इज दैट कि नंबर में यूज़ होना चाहिए, नंबर में हमें कल्चर से सेल्स अवेलेबल होने चाहिए फॉर ट्रांसप्लांटेशन. जो एक डोज होती है ट्रांसप्लांटेशन की, उससे कम सेल्स होंगे तो ट्रांसप्लांटेशन नहीं हो सकता, तो उसको नंबर में पूरा होना चाहिए. एंड एज द यूनिवर्सल sample of collection, urine derived cells can serve as an outstanding option for treatment of disease. ये नॉन इन्वेसिव है, इसमें कोई बॉडी में सूचर नहीं लगाना पड़ता, कुछ भी नहीं करना पड़ता. सो अगर ये सक्सेसफुली हो जाए तो दिस इज़ द बेस्ट ऑप्शन for production of stem cells. And with more extensive study, urine cells can be employed as a preservation method for patient cells to serve as a cell bank for therapy and disease modeling study by generating iPSCs. जो फिगर में दिखाया था कि induced pluripotent cells अब बना के फर्स्ट इसमें कन्वर्ट कर लिया, देन उसको फर्दर डिफ्रेंशिएट कर दिया इन एनी काइंड ऑफ सेल. तो सेल बैंकिंग अगर हो सकती है तो इट इज़ पॉसिबल टू जनरेट iPSCs, एंड देन लेटर ऑन जब भी ज़रूरत है उनको उस किसी पर्टिकुलर सेल लाइन में कन्वर्ट किया जा सकता है या डिफ्रेंशिएट किया जा सकता है. In addition, urine cells can also serve as a replacement for current MSCs. mesenchymal cells से इनको रिप्लेस किया जा सकता है अगर ये सक्सेसफुली डिफरेंट वेराइटी के सेल्स बनाते हैं, यानी कि मल्टीपोटेंट है. While MSC is derived through invasive methods such as bone marrow harvest or liposuction. Bone marrow harvest or liposuction is the most popular techniques or sites. Bone marrow साइट हुआ और लिपोसक्शन एज अ टेक्निक. तो मतलब ये है कि बोन मैरो से अभी MSCs डिराइव करने के यूज़ करने के लिए लिए जाते हैं या लिपोसक्शन करवाया जाता है, वहाँ से मिलते हैं. तो टू जनरेट अ डोनर बायोबैंक, तो MSCs के लिए जो बायोबैंक है उसमें इन्वेसिव प्रोसीजर्स यूज़ होने पड़ते हैं. However, this may require further expandability and qualitative studies, especially with the recent report of considerable difference in isolation, efficiencies and cell culture issues. जो यूरिन सोर्स है उससे जो सेल्स मिल रहे हैं उनकी क्वालिटीज़ डिफर करती हैं वन बैच टू अनदर, और सेल की ग्रोथ होने में भी दिक्कत आती है. दैट इज़ वाई देयर इज़ नीड ऑफ मोर रिसर्च इन दिस फील्ड. नाउ कंसर्न्स क्या हैं, आखिर क्यों नहीं ईजी है अभी, या इट रिक्वायर्स मोर एंड मोर रिसर्च बिफोर इट कैन बी used for regenerative therapy. So efficacy of the cells in terms of functional differentiation and long term transplantation के लिए इनको further टेस्ट करना पड़ेगा और रिसर्च करनी पड़ेगी, and there is need to check the immune reaction after transplantation. Then a significant intervariability in the expression of reported markers and the reasons for variation need to be addressed. One possible explanation for this heterogeneity in terms of gene expression can be due to mixed nature of isolated cells. तो होमोजीनियसली एक्सक्रीट आउट नहीं होते यूरिन सेल्स, heterogeneity मिलती है, and that is why we get variable markers. अगर एक सोर्स से सेल्स लिए गए और वहाँ पे कोई मार्कर्स के एक्सप्रेशन की एक्सपेक्ट किया जा रहा है कि वो मार्कर्स आएंगे, वहाँ पे मिलेंगे, तो ऐसा हो सकता है कि वो बदल गए, और उसका रीज़न यही है कि मिक्स नेचर है और बहुत ज़्यादा सेल्स वेरी कर सकते हैं. This is also supportive of the fact that human urine contains complex constituents and the cells removed from the body. तो ये ऐसा इसलिए होता है क्योंकि डिफरेंट पार्ट से आए हुए सेल्स मिलते हैं यूरिन में. Sorting of isolated urine cells at early passages can minimize the variation arising from heterogeneous nature of urine cell cultures. यूरिन का सैंपल जब भी लिया जाता है, मॉर्निंग की फर्स्ट यूरिन को प्रेफर किया जाता है. तो अगर उस समय का यूरिन ले तो इट इज़ पॉसिबल कि वहाँ पे वेरिएशन heterogeneous नेचर के नहीं मिलेंगे. For isolation and cell culture, urine storage conditions after collection must be standardized. अभी again research से वो भी standardize करना है कि कौन सा सीरम सैंपल यूज़ होना है, नहीं होना, कल्चर मीडियम क्या है, what are the temperatures, most appropriate temperatures, and how much time will it take. तो ये सारी बातें अभी भी इस रिसर्च की स्टेज में है. Then the differences in gene expression of urine cell markers in different samples obtained from same individual at different times. Intravariability, अगर एक ही इंडिविजुअल है और वो दिन भर में कई बार उससे सैंपल लिया था, अगेन मार्कर्स का एक्सप्रेशन अलग है, उनका जीन एक्सप्रेशन ड्यूरिंग द डे टाइम अलग पाया जा रहा है. तो उसके कॉजेज को चेक करके एक होमोजीनियस पॉपुलेशन ऑब्टेन करना अभी भी एक कंसर्न है और रिसर्च का मटेरियल है. There is need to investigate if urine cell generation will be influenced by diet and medication. Okay, this part is not much studied कि क्या होता है, कौन सी डाइट खाने से क्या उसकी वजह से होमोजीनियस सेल्स नहीं मिलते हैं. Generating clinically graded autologous cells from urine sample for cell therapy still needs comprehensive preclinical analysis to generate a standardized procedure. So, at last, conclusively, with detailed testing and optimization, urine cells can serve as the future of regenerative therapy in terms of generating quantitative and qualitative cells for both organ reconstruction and to understand pathological mechanisms using disease modeling. डिजीज़ मॉडलिंग माने कि डिजीज़ प्रोड्यूस की जाए और फिर उस पर उसकी डिफरेंट फिजियोलॉजी और उसकी प्रॉपर्टीज़, डिजीज़ की प्रॉपर्टीज़, डिजीज़ को ट्रीट करने पे क्या होता है, उसकी पॉसिबिलिटी, ये सारी चीज़ें टेस्ट करने के लिए विट्रो टेस्टिंग के लिए अभी भी कंडीशंस स्टैंडर्डाइज होनी है. Now, these are the references which I I have gone through for understanding this topic. This is also one important paper, and thank you. If you like my lecture, please subscribe it, like it, and share it, and keep watching my lectures. Thank you.